how you put a SIM card in a new phone and suddenly it becomes a personalized device that knows all of your song preferences and previous locations? Well, what if you could put a chip in your brain to achieve the same effect? Okay, maybe that's terrifying, but while it's not something that's done in humans, that technology might be closer than you think. So today, let's talk about mind control and some real-life ways we can make brains do what we want, whether it's to learn more about them or to help people with certain medical conditions live life to the fullest. So let's start with the somewhat checkered history of electrical therapy and how it's become an effective treatment for epilepsy, Parkinson's, and other conditions.
It's a classic sci-fi plot device. A chip is implanted into someone's brain, and with a jolt of electricity, their brain is suddenly under someone else's control. As with all good sci-fi, this is mostly fiction, but there is a bit of real science in there. Obviously, you can't just plant a chip in someone else's head and start manipulating their thoughts and behavior. But doctors and scientists can use electricity to activate or inhibit certain parts of the brain to help treat serious medical conditions or mental disorders. And these treatments have changed the lives of many patients, especially those who may not have responded well to other treatments like medication or psychotherapy. Using electricity as a medical treatment is not new. We've known for centuries that electricity can have an effect on brain and nerve function, but for a long time we did not understand why. For example, ancient Romans were known to actually use an electric fish, a kind of ray known as torpedo nobiliana, to treat patients with a variety of symptoms from headaches to seizures. Just put the fish on you. The ray was known to stun anything that came into contact with it, so medical professionals of the time would just put this ray on the heads of their patients to numb their symptoms. Put a fish on it! How do we know that they didn't know why this worked? Because sometimes they used the same technique to treat hemorrhoids. Just put a fish on it! Fast forward to the 19th century when physicians started experimenting with applying electricity to the exposed human brain. One physician even experimented on the exposed cerebral cortices of decapitated prisoners. He found that by stimulating the surface of the cortex with electricity, he could cause the face to actually grimace. Gruesome as it was, this at least provided some new scientific insights. It proved that the brain could be directly stimulated with electricity. Scientists were starting to learn a very important truth. The human nervous system relies on electricity to function. Nerve cells use electricity and chemicals to communicate with one another, transmitting information like little messages throughout your body and brain. Your body depends on these electrical signals to do just about everything. From smiling to walking, processing memories or feelings, it all comes back to electrical signals in the brain. Beginning in the 1930s, physicians built on this knowledge, developing something known as the Montreal procedure to help treat epilepsy patients. This method used an electrical probe to directly stimulate different parts of a patient's cerebral cortex. After removing a section of a patient's skull, a physician would use the probe to electrically trigger different sections of the brain. While the patient was awake and providing feedback, the physician would use the probe to stimulate different parts of the brain, which would produce sensations like flashing lights or smells that the patient could report. Epileptic patients sometimes experience auras before the onset of a seizure. These auras might involve very particular sensations or perceptions, like the smell of burnt toast, for example. If a patient reported something similar to their seizure aura as a result of the electrical stimulation, the doctors knew they'd found the part of the brain causing the seizures. And then they could remove or destroy a small section of tissue there. Freaky as it sounds, this often successfully reduced the patient's risk of seizures. And the Montreal procedure was also used to map out the brain, notably the sensory and motor cortices. By the second half of the 20th century, physicians were learning that certain types of electrical stimulation were especially useful in treating symptoms related to neurological movement disorders like Parkinson's disease. Though it's still not fully understood, it's believed that electrical stimulation affects the way nerve cells in the brain, called neurons, communicate with one another. For example, deep brain stimulation, or DBS for short, is often used to treat symptoms associated with Parkinson's disease. Electrodes are planted deep within the patient's brain. These electrodes release controlled electrical signals, and these messages may encourage the brain to release the energy-carrying molecule that we all use, ATP. As the brain releases ATP, a chemical called adenosine gradually builds up, initiating a process that helps slow nerve signals related to uncontrollable movements like tremors. Though it was originally approved to help treat Parkinson's disease, DBS may also help with treating symptoms related to depression or obsessive-compulsive disorder, since DBS can help with regulating brain activity. By the 60s and 70s, researchers were starting to experiment with electrical implants within the brain to help treat chronic pain, epilepsy, and other neurological disorders. But most of these implants weren't practical because they needed bulky external power sources. However, the invention of the cardiac pacemaker in the 1960s showed that it was possible to place a mobile power supply into a person's body safely. With DBS currently, for example, the electrodes planted in a patient's brain are controlled by a small power source that's placed in a patient's chest. Today, a number of different brain stimulation therapies are used to help treat a variety of disorders. Aside from DBS, there's also electroconvulsive therapy, or ECT. This method is used to treat disorders like depression and schizophrenia. With ECT, an electrical current passes through electrodes on a patient's scalp, triggering the brain to cause a brief seizure. We're still not exactly sure how ECT works in the brain, but it might involve changes in both neurotransmitters like dopamine and the brain's stress response. Likewise, a technique known as vagus nerve stimulation uses electrical pulses to stimulate the parts of the brain that are associated with seizures to treat epilepsy. It can also be used in other cases like severe depression. In this case, the pulses come from a device implanted in the patient's chest and stimulate the left vagus nerve, which passes a signal onto the brain. This nerve is part of the control system that helps the brain regulate involuntary actions like heart rate. It also acts like a freeway of sorts, sharing information from the brain to the heart, abdomen, lungs, and neck and back again. These pulses may help control or limit the effects of seizures by spurring neurons to release neurotransmitters that help to modify the excitability of certain cells, lessening or even stopping seizures. Electrical brain implants might sound like science fiction, but these techniques are grounded in pure reality. Brain stimulation techniques have radically improved how physicians treat neurological and mental health conditions. And as these technologies improve, neuroscientists will hopefully continue to develop more effective and less invasive treatments, truly changing the lives of countless people around the world.
Your brain sends electrical signals from cell to cell. That's part of what allows you to think and move and do pretty much everything you do. So it actually follows pretty logically to use electricity to help treat it. But how many other ways are there to alter the electricity in your brain? Turns out, a lot.
People around the world are strapping batteries to their heads to supercharge their brains. Seriously? This is a thing. It's called transcranial direct current stimulation, or TDCS, and it involves using electric currents to stimulate your brain. The companies that want to sell you TDCS devices claim that it has all kinds of benefits. They might say that it'll boost your memory and focus. Some even claim that it can do everything from treat depression to help you sleep better. If that sounds too good to be true, it is. The truth is a lot more complicated. Studies have found that TDCS might help with some things, but there's also plenty of controversy about it among scientists, and the jury is still out about how exactly TDCS affects the brain. Even though it involves electric currents, TDCS is a lot less intense than the kind of supercharged electric shocks used in electroconvulsive therapy. That's the more famous electricity-related therapy that's sometimes used to treat things like really severe depression. A TDCS headset contains two electrodes called the anode and the cathode. The idea is that the anode will boost the area of the brain beneath it, whereas the cathode will inhibit the area beneath it. The choice of those areas, of course, depends on the particular functions you're looking to play with. Once everything is in place, a small electrical current of around 0.5 to 2.0 milliamps is sent through the electrodes right into your head. Since the current is so small, it's not painful as long as you set it up properly. The most people tend to feel from this kind of current is a mild tingling sensation on their scalp. A TDCS session typically lasts around 20 to 30 minutes, and people usually do a number of sessions over the course of a few weeks. The thing is, most people doing this at home might be making their scalps tingle for nothing, because we still aren't sure if TDCS actually does a lot of the things companies selling these devices are claiming. Entering the world of TDCS experimentation has a pretty low bar since headsets are fairly inexpensive. Unfortunately, that's made the literature a little messy. One problem is that we still aren't totally clear on what TDCS does to the brain on a physiological level. We do know that current can affect the way neurons fire, and researchers think that the mild current from the electrodes changes the membrane potentials of neurons, allowing them to fire more easily. The idea is that with enough stimulation and repeated exposure, the neurons that are getting the stimulation will start firing more. Over time, that might nudge damaged or underperforming brain areas toward functioning in a healthier way all by themselves. But this is all still the subject of some debate. One study published in 2016 used a cadaver to measure how much current from TDCS managed to travel through the skull to the brain. Electrodes placed on the inside of the skull showed that around 90% of the electricity applied through TDCS didn't get through at all. That study raised a few eyebrows, but according to a lot of TDCS experts, the findings actually made sense since live tissue is better at conducting electricity. Plus, there's a lot of evidence that TDCS affects brain function, so there must be some amount of current, however small, making it through. Which brings us to the other main problem when it comes to TDCS. We still don't exactly know what effect it has. A lot of TDCS studies are small pilot studies that need to be replicated before we can draw any conclusions from their results. And some aren't so tight in the methodology department. In meta-analyses that look at results across multiple TDCS studies, researchers have pointed out that the methods used in different studies are so inconsistent that it can be hard to compare them. There is some well-designed TDCS research out there, though, and based on those studies, researchers have found that it might be an effective treatment for mood disorders, psychosis, and dementia. It might even help people recover from strokes. A 2016 meta-analysis of 17 studies found that stroke patients who did TDCS treatments regained more motor function than patients in the control groups who did fake TDCS treatments. Scientists are also looking into TDCS as an alternative treatment for depression. A meta-analysis of data from 6 randomized control trials with 289 patients in all showed that TDCS relieved symptoms of depression even in people who'd felt no benefits from medication. But that's the thing. Researchers are still looking into all of this. TDCS seems like it could be really promising, but there's a lot that we still don't know. Sure, some studies have found that TDCS helps stroke patients recover their motor function, but that doesn't necessarily mean that it will give healthy people better motor function in general. Plus, these studies are done in a clinical setting, which is totally different from buying a device and giving yourself a TDCS treatment at home. If you put the electrodes just a couple centimeters off from the right spot, or the current is slightly lower or higher than it should be, you might end up stimulating your brain in a way that's completely different from what you wanted. So there is some solid evidence that seems to point to TDCS being a useful therapeutic tool in the future. Just maybe don't run electric currents through your brain on your own until we know a little more.
Some of the brain manipulations that scientists use regularly in rodents aren't ready for use in humans yet, if they ever will be. Right now, the role of this technology is helping researchers study the brain. To shed light on how the brain works, some researchers literally shine a light on the brain to manipulate it. Intrigued? Here's how it works.
You hear people make comparisons all the time about how your brain is like one big, ultra-sophisticated computer, and that's fine, but the idea really only goes so far. Because, you can't really program your brain like you can your desktop, which is kind of a bummer. I want to know Kung Fu. And if we could just get in there and start manipulating neurons, we could solve a lot of the struggles our brains and bodies cause. But what if we could do that? Recently, scientists working with mice have been able to send new signals into their brains, ones that seemed so natural, the mice may have no idea they weren't their own. It seems a little sci-fi, but it's real research, and someday it could lead to some pretty amazing new treatments. These studies center around a branch of neuroscientific research called optogenetics, which makes use of light-sensitive proteins inserted into neurons through genetic manipulation. Once the proteins are there, researchers can control the firing of those neurons using just a flash of light. We actually did a whole episode on the mechanics and history of optogenetics that we'll link to after this, but the general idea is that this method alters what certain receptors on cells respond to. Instead of waiting for a chemical neurotransmitter to tell them to do their job, an altered receptor will take the command from a flash of light transmitted through a wire. When optogenetics first entered the scientific literature in 2005, the initial technique involved flooding a relatively large area of the brain with light to activate it. This is still generally the way it's done for now, and it does work. With it, we've been able to get groups of neurons to fire and even induce behavioral changes in rodents. Like one experiment used optogenetics to get rats to walk in circles, which is pretty amazing. Also a little upsetting.
Why is it I have to do this now. This is my life. I'm a rat.
But one of the downsides of this method is that lighting up a whole area can be imprecise. Trying to tease apart what's happening with the specific neurons you want to study versus what's coming from ones lit up in the crossfire can be pretty darn tricky. However, the times, they are a-changin'. Thanks to advances in tech and biology, a group of scientists recently figured out a way to target specific individual neurons rather than a relatively large patch of tissue. They published their results in April 2018 in the journal Nature. And brace yourself for some sci-fi, because they're doing it using holograms. Visual holograms, the kind you see in Star Trek or Star Wars, use multiple beams of light to project an image. But holography in general is really just about light interference, and that's what this technique relies on. The team called their method 3D shot, which stands for three-dimensional scanless holographic optogenetics with temporal focusing, which I'm glad they have such a good acronym for that. Unlike the original optogenetics technique, 3D shot focuses light from several sources on individual target neurons. Only the specific neurons in the tiny area all the light crosses receive enough light to activate. That can eliminate a lot of unintended firing, so these results are much less muddy. And it seems effective. Scientists using this method have already observed mice brains firing at a natural timing and frequency when stimulated. They can tell based on the amount of calcium the cells are using, which is a good estimate for the rate of neural firing. And because that firing appears to really closely mimic the way their brains would naturally detect and process stimuli, it's likely that to those mice, any experiences induced using 3D shot would be pretty realistic. I mean, unfortunately, we cannot ask the mice about this, but if we can stimulate their brains into firing in the same patterns as it would when, say, seeing, feeling, or smelling something real, that seems reasonable. One way we can test this hypothesis is by observing actual behavioral changes, like a flick of a switch causing a mouse to stop in its tracks, which we haven't done yet. But the good news is those experiments are currently underway, so we'll keep you updated. If scientists are able to make neurons fire realistically, though, and if those experiences feel real even though they're not, well, in theory, this technology could eventually be used to alter, memories by editing the way neurons associated with those events fire. We could make mice perceive things that aren't really there, all with the flash of a few lights. We could download kung fu into their brain, like the Matrix. Finally, rats doing kung fu. That might be getting a little ahead of ourselves, but honestly, maybe not as far as you'd think. The research team responsible for 3D shot already have a few ideas about how they're going to sort of copy and paste brain activity to achieve something like this. If they can measure the sequences of activations in specific neurons involved in the behavior they want to program, 3D shot could feasibly send that pattern into a different brain. Basically, programming it. Brain programming. Now, even if we're making great progress with mice, applications in humans are likely a very long way off. Optogenetic methods require specific light-sensitive proteins to be placed into the genome, which could get risky. But if we could do it safely, that would be hugely useful, not only for research, it would be for research, but also for future treatment options. Think about it, if we could design a prosthetic eye that picked up visuals from the world and transformed them into the right patterns of neural firing, you might be able to restore someone's vision. That's just one out of thousands of examples, but we have a lot of work to do before we can get there. So I'm sorry if you wanted to download very good dancing skills. I also want that to happen to me, but it's gonna be a while. Until then, maybe we should take some classes until the research is a little further along.
Optogenetics is pretty sophisticated technology. There's a lot that goes into its development and use. So here's a basic user guide to mind control by light.
Imagine being able to remote control your cat. By clicking a remote at a receiver on your beloved kitty's head, you could make it walk, turn in circles, or even stop moving altogether. Maybe with the press of a button, you could even put it to sleep or turn off its sense of pain when it hurt itself. And even weirder, what if you could control your own body that way? Well, there's a scientific technique that might someday deliver just that. It's called optogenetics, and it's a method for controlling the function of cells using light. Even though it sounds a lot like mind control, optogenetics is really most useful useful for learning what specific cells do or for treating certain conditions. It does this by using light to control certain pathways on a cell's surface called ion channel receptors. These are sort of like switches that start and stop electrical signals traveling down your cells. Normally, they're activated when molecules like neurotransmitters attach themselves to the receptor. That causes charged atoms, or ions, to move into the cell, which generates an electrical current that can make other cells stop or start firing. These channels are ultimately what makes your body move and function. Optogenetics works by using light to control this process instead of molecules like neurotransmitters. Using fiber optic wires, scientists can transmit precise light pulses that last just 1/1000th of a second directly to a small group of cells, and that triggers ion channels to open and start sending signals. Now, even before this method, researchers have been trying to control ion channels for years, since that could help us figure out how cells work or even treat some diseases. But other methods, like drugs or electrodes, tend to be too slow or imprecise. To really study a small number of neurons, researchers needed fast, accurate signals, and that's where optogenetics shined a light on the problem, literally. It all works thanks to special proteins called opsins, which are naturally found in organisms like microbes or green algae. When they're exposed to certain particles of light, they'll generate an electrical signal and open a cell's ion channels. We've actually known this about opsins since the 1970s, when researchers noticed that one in certain bacteria, called bacteriorhodopsin, opened its ion channels in response to green light. And today we know about plenty of others, which start and stop firing neurons in response to all kinds of light. But no matter how many opsins we found, it took until the beginning of the 21st century for scientists to really understand the applications of them. They realized that if they could somehow get these opsins into animal cells, they'd be able to control the ion channels in the fast, accurate way they needed. And in 2005, they did it for the first time. In the journal Nature Neuroscience, researchers announced that they'd introduced opsins into a rat's brain cell, although the cell was in a petri dish and not a live animal. When they shone blue light on it, it showed a spike in electrical activity. Essentially, they had made a light-sensitive brain cell. But trying this on live mammals was a lot harder, because smuggling opsins into a living cell is tricky business. To do it, researchers had to develop a special virus that could transfer the protein onto the surface of an animal's cells without the virus itself running haywire. Then if that worked, they could just insert a wire into the animal's brain and use an LED or laser to start manipulating neurons. And they did it. In 2007, scientists demonstrated this technique for the first time in a live animal by applying optogenetics to cells in the motor cortex of a mouse. By transmitting blue light down the optical fiber in the mouse's brain, they could make the mouse walk in circles and make it stop when they turned off the light. Pretty weird. Many other studies also use optogenetics to study how cell activity correlates with behavior and bodily function. For example, in one study from the journal Nature, the researchers manipulated cells that put fruit flies to sleep and then could wake them up on command. Those same cells are involved in the fruit fly's internal sleep clock, which has similarities to the one in humans. In mice, researchers have also used optogenetics to study behavior related to hunger, which could help us model obesity in people. It even has a role in helping us understand and maybe someday treat certain diseases. Like back in 2011 in the journal Molecular Therapy, researchers claimed they'd used optogenetics to restore light sensitivity to cells in a mouse that had lost its vision. That could someday help develop human treatments for a disease called retinitis pigmentosa, which destroys light-sensitive cells in the retina and causes blindness. And another study from the journal Neuron pinned down specific mice neurons involved with motor control. In humans, cells in a similar part of the brain are affected by Parkinson's disease. Now, even though mice, rats, and fruit flies have brain structures with some features in common with humans, our brains are way more complicated to understand. We'll need a lot more work before we're ready to give people remote controls for their brains. Still, it's possible that someday, tiny optogenetic devices will be working away in our bodies, offering targeted therapies and cyborg-style nerve implants. We're definitely not there yet, but if it ever happens, just be careful not to leave the controls to your body lying around the house. If your cat sits on it, it might end up controlling you instead. Although really, I guess that's not much different from owning a cat today anyways.
Optogenetics really does allow scientists to control an animal's brain and make changes happen almost immediately. But it's not the only tool in their toolbox. They can use different approaches to solve different problems. So for example, let's say you want to improve your memory. Scientists have figured out how to do that too.
When you want to remember where you left your keys, you will hopefully conjure up a sort of image or idea of where you last had them. While it may seem like that memory just magically appears in your mind's eye, the truth is that memories are far more physical. That means they have to live somewhere in your brain. But like where? Is there a shelf labeled keys right next to the one that says, "That time I tripped and fell down during the school play"? Well, it's definitely not that literal, but thanks to advances in neuroscience, we can finally pinpoint the actual location of a memory, at least if you're a mouse. To find a memory, first you have to make one. And it's easy to do that. You just do something. Anything, just like, watch me do this. That's in your brain now. You're stuck with it. Your brain will then pick a bunch of cells to store a memory of the thing you just experienced, and it will activate them all at the same time. But those cells aren't all in the same place, because the parts in your brain specialize in different things. Your brain will pick cells across regions to store all the different aspects of a memory. For example, neurons in the visual cortex will store what you were seeing, and the cells in your amygdala will store how you were feeling. That specific pattern of cells all firing together is what the memory is now, in all of its multisensory glory. The word scientists use to talk about this physical trace of a memory in a brain, the pattern of cells that activate to recall a memory, is an engram. It's the unique pattern of cells activating together across the brain that makes a memory. And these cells are actually changed by the learning experience. They form stronger connections with each other than with other neurons, and they develop more dendritic spines, protrusions that help neurons talk to each other. So, how do you find an engram? Ideally, if you could watch neurons fire, you should see the same ones fire when the same memory is activated, and we can do that in mice. In a 2007 study published in the journal Science, researchers taught a mouse to associate a mild foot shock with a sound. This is a pretty commonly used protocol for studying memory. If you see the mouse tense up, you know it remembers a shock is coming. And don't worry, it's like a little zap. The researchers used a special glowing protein to tag the neurons that fired as the mouse learned. That let them monitor what neurons fired when the mouse heard the tone and remembered what was coming next. A few days later, when the researchers played the sound again, they saw the same neurons firing. Basically, they saw that memory. Now, the next question was whether altering the neurons in the engram would mess with the memory, which would prove that those neurons were holding that memory and could even lead to, memory editing. So in a 2009 study, also in Science, researchers infected an area of the mouse's brain with a virus that increased both how readily those neurons would activate and the number of those neuron-connecting dendritic spines. The virus basically made this area full of superneurons, which the researchers hoped would be very likely to be used for storing a memory. Then the researchers once again taught a mouse to associate between a sound and a foot shock, and then, just as planned, the superneurons were used to store that memory. But the virus also had a kill switch, which the researchers activated after training. Like a tiny viral assassin, it took out the superneurons. When they played the sound again, the mouse didn't freeze up. It seemed to have no idea that a shock was coming. It appeared that killing those specific cells also killed the memory. In subsequent research, scientists were able to manipulate both real and false memories. In one 2012 study, the researchers used a technique called optogenetics, where by shining a special light on a part of the brain primed with light-sensitive proteins, you can turn specific neurons on. The researchers taught a mouse to associate a sound with a shock, and then pumped the engram cells, where they knew that memory lived, full of this light-sensitive protein. When they turned on the light, the mouse acted like it had just heard the sound and expected a shock, even though there was no sound. And in a 2013 study published in Science, researchers moved a mouse from an old cage to a new cage and trained it to fear that new cage with electric shocks. Then they used similar optogenetic techniques to make the mouse remember its old cage while it was in the new one. When they put the mouse back in its old cage, it still seemed frightened. It thought that was where the shock had happened. These findings might make it seem like neuroscientists are officially masters of memory, able to edit, implant, and delete memories at a whim, but it is very early days here. We're nowhere close to handheld memory-wiping gadgets a la Men in Black. But if manipulating engram cells can add, delete, and modify memories in predictable ways, that does mean engrams are a useful model for studying memory. Memory is such a crucial part of our lives and our identities, and by better understanding how it works physically, we demystify it. That could open the door for better treatments for conditions like Alzheimer's or post-traumatic post-traumatic stress disorder. So while we will probably never have zappers to remind us where our keys went, we are getting closer to understanding how our brains store the memories that make us us.
So we know there's a lot of ways to control the mind, but what happens when you don't have control?
You know those closed door buttons in the elevators? The one that you hit over and over again because the door's still open and you're like, can we go up, please? And you kind of suspect that they don't actually do anything. Well, it probably doesn't and hasn't for a while. As part of the Americans with Disabilities Act of 1990, elevator doors are required to stay open long enough for someone in a wheelchair or with trouble walking to get on, and no button is allowed to make that shorter. Firefighters can override it, but for the rest of us, it's a fake. Our lives are full of other non-working buttons too. When cities switched over to using computers to time traffic signals, that meant many crosswalk buttons became shams for at least part of the day. These buttons might be a little frustrating, but it's hard not to push them, and the lie isn't all bad. In a small way, these placebos might even make you feel better. It comes down to something psychologists call perceived control. Perceived control is how much control you think you have over a situation. Sometimes your estimate is about right, or sometimes it's more or less than what you actually have. But in general, studies suggest that thinking you have some control in life is a really big deal for physical and mental health. Nursing home residents, for example, have been found to be happier, more active, and more alert when they're encouraged to make their own choices. Employees are happier, healthier, and less likely to quit when they feel that they have some control over their schedules and job duties, and people with panic disorders are less likely to have an attack when they feel they're in control. In one experiment, panic disorder patients were hooked up to an air mask that provided oxygen with a bit of extra carbon dioxide. They were told that they could lower the amount of CO2 by turning a dial that would light up if it was working. Half the patients had dials that lit up, half had dials that did not. In reality, none of the dials did anything, but the group that thought it was functional had fewer symptoms of anxiety. And the amazing thing is, in many cases, there is no difference in the amount of real control someone has. None of the panic attack patients could change anything. What matters is the perception of control. Which brings us back to those elevator or crosswalk buttons. Can we really not tell when we're not in control? It turns out people are remarkably good at deluding themselves. Psychologist Ellen Langer uncovered this tendency back in the mid-1970s when she set up a series of gambling experiments. In one, office workers were either given a lottery ticket or allowed to select one from a pile. Then they were asked how much they'd sell their ticket for. Even though the ticket they got was totally random, participants who got to pick their tickets for themselves thought that they were worth more and said they'd sell their tickets for higher prices. In another study, college students placed bets on whether a playing card pulled from the top of the deck would be higher than their opponent's. They could wager up to a quarter. It was the 70s after all. When students were matched with a well-dressed, confident opponent who was in on the experiment, they bet less. And when they were matched with a shy, awkward person who also turned out to be an actor, they bet more. In both cases, everything was determined by chance. There was no way to affect the game, but participants acted as though they could. Langer called this irrationally high sense of agency the illusion of control. It's just when you think you have more control than you actually do. Exactly why we do this isn't clear, but our sense of being in control isn't a strict representation of reality, it's a perception, something actively constructed by our brains. And it doesn't take much for us to link things together. So when it comes down to it, every time you push that elevator button, even if it feels like an eternity standing there with a bunch of strangers, the door does always close. It feels like you were in charge. And so if it makes you feel better, go ahead and keep pushing that fake button. For a jolt of real power, though, pound that open door button. That one still works.
So there you have it. We do have the technology to control brains to some extent. And while most of this work was done in mice, there is potential for specific clinical treatments in humans. Maybe even better news is that you don't even need to control other people's minds. Learning more about how to control your own mind is a feat all on its own. Thank you for watching. Today is my last day hosting for SciShow Psych, so thank you so much for joining me over the last few years as we've explored psychology and brains and humans and all of this exciting stuff together. If you'd like to follow along with me on my next adventures, you can find me on Twitter at Brit Garner, and you can also check out my other content at youtube.com/natureleague. Thanks so much.